Okay, everybody, this is Hormones, Chapter 18. We are skipping the neuro stuff to ensure that we get through some of these hormones. So Test 3 in Bio 207 with Dr. Granier is going to be Chapters 10 and Chapters 18. So it's all the muscle and all of the hormones. Okay? So the hormones come from the organ system known as the endocrine system. Now, as we've divided out and figured out more about proteins made in different types of cells and different organs, we have found that a lot of molecules can actually be classified as a hormone. So this organ system is basically going to be intensely focused on the classical endocrine system. And the classical endocrine system is defined in two ways. One, it's typically some type of epithelial gland. So epithelial glandular tissue that produces some type of molecule that is traveling from that glandular tissue to a target organ to have their effect. And the key here is the molecule that's going to leave the gland and travel to the target is going to do so in the bloodstream. So it's the classical endocrine system is typically the originating material is an epithelial glandular organ. The molecule made is going to travel in the bloodstream from the glandular origin to a target to have their effect. Okay? So when you look at the endocrine system, the main original glands are the pituitary gland in the brain, okay? And what we'll learn is that the pituitary gland is heavily influenced by nervous tissue as the hypothalamus, the part of the brain in the diencephalon that is going to influence the pituitary gland. And part of the pituitary gland is actually extensions of neurons from the hypothalamus. So these two are very interrelated. But the gland, the pituitary gland, is the epithelial tissue that kind of fits that classical equation of gland producing molecules that travel in the bloodstream. Okay? All right. The thyroid gland is our second typical gland producing molecules traveling in the bloodstream. The adrenal glands, suprarenal glands, meaning they're on top of the kidney, is the third. The pancreas is the fourth. All right? Pineal gland in the brain is our fifth. Parathyroid glands, which sometimes get grouped in with the thyroid gland, sixth. And then your gonads, which the ovaries and the testes, technically speaking, are epithelial tissue. So those are the common glands that produce molecules that travel in the bloodstream and have effects on their target tissues. Now, they're not the only organs that produce molecules that travel in the bloodstream and have effect elsewhere. And so these other items actually are going to have hormones because they're going to make items that travel in the bloodstream and have effects outside of the target or outside of the initial origin tissue, but they're not, technically speaking, all the time epithelial glandular systems. So they don't fall into the classical endocrine system. And so we might not cover them that much in this chapter because the heart is a cardiovascular muscular organ. And so we will mention briefly one of the hormones it makes, but we're going to do that more so in Chapter 21, Chapter 26, where the kidneys come into play. All right? The thymus gland is actually a gland that has nothing to do with making molecules that signal other particles. It's where red, white blood cells come to mature. So it's a gland, but it doesn't actually make hormones that travel much in the bloodstream. We are going to talk about adipose tissue because we don't really have another opportunity to do so. But technically speaking, adipose tissue is a connective tissue. So it's not technically epithelial glandular tissue, but it does make some molecules that travel in the bloodstream to go to the stomach, that go to the GI tract, communicate there, communicate with the liver, and communicate with the brain in terms of fat production, fat regulation, and appetite stimulation and suppression. The digestive tract, that could be a whole endocrine system unto itself. And so they are a variety of hormones that communicate between the organs so they know when food is coming downstream or to communicate upstream to decrease or increase secretions of enzymes. So there's a lot of organs, and we're still just, you know, scratching the surface there, because some of the hormones not only communicate within the small intestine, large intestine, stomach, but they also go to the brain to influence appetite, hunger, and suppression. And then we've already talked about EPO from the kidneys and calcitriol last semester with the bone, but we'll hit that again, okay? So most of what we're going to do in this chapter is hit the classical endocrine systems and sprinkle in a few extra things that we don't really have an opportunity to talk about anywhere else. Okay. All right, so again, this idea of a hormone. A hormone is a molecule that is going to leave a cell, travel in the bloodstream to a target, and have their effects. So there are other types of communication in the body. And so just for review, one type of communication is when cells are connecting through each other. And this happens in the heart tissue. This happens in smooth muscle. Wherever cells are physically touching each other, what happens to one cell, because they are physically touching and connecting, is going to happen to the other cell. And that is a very fast way to communicate between cells, and it's how our hearts beats basically in unison, or how smooth muscle cells in a region can coordinate and constrict in a certain area together. All right, it's very effective, but again, you have to have the cells physically touching. And if the cells are physically touching and connected to each other, it's very hard to get neurons in between those cells, very hard to get capillaries in between those cells. So it's very hard to deliver other messages. It's very hard to deliver oxygen. So it's not the most ideal communication for many areas of our body. So the second type of communication, which we're going to talk a lot in the immune system, and again, this comes into play with how cells that are near but not touching the cells around them can communicate to them. All right, and so this is how cells that are in distress can communicate to the cells around it that that some virus or pathogen is trying to hijack them, and they can help the other cells, you know that there's a threat in the area, so they can beef up their security measures. This is how the cells communicate to floating and free macrophages and other white blood cells to come over and help them with a defense. So there's a lot of cytokines is typically what we call these paracrine communication factors that are released by a cell that will travel locally or not very far from the originating cell, and they communicate with lots of different types of cells. So there's not a specific target all the time. Sometimes there is, like white blood cells, but it can also communicate to any and every cell that the molecule comes into contact with. And so some of the categories of paracrine communications are interleukins, so you sometimes see them as IL proteins, interferons, complements, prostaglandins, and those are fat-based molecules, leukotrienes, which sometimes for some of the asthma medications they talk about, or allergy medications, there are leukotriene, suppressors. Echinosoids. So there's some different molecules that kind of come into play here that are communicating ability with lots of different cells. All right. The endocrine communication, again, it's some type of, classically speaking, glandular cells, so epithelial cells, making molecules in response to some stimulus that allows these molecules to travel in the bloodstream to a downstream target tissue and have an effect there. Now, because the molecules travel in the bloodstream, their effects are not instantaneous. So paracrine and direct communication may have effects that are pretty fast-acting because the molecule doesn't have a long distance to travel. But in hormones, because they have to travel in the bloodstream, and the bloodstream is, it's not a guided tour. It's kind of a luck of the draw that you end up at your organ. So it might be that you have to go through the heart maybe 3 or 4 times before you end up on the right little forks and pathways to get to the target organ. All right? And so there's more time that is going to be needed before effects are seen. So that's part of the reason why endocrine communication is going to control slower changing items like growth, reproductive cycles and seasonal cycles, because again, we want the growth, we want the changes to happen in a more coordinated, slower controlled fashion, and having the molecule with a longer lifespan with a change that cannot necessarily happen instantaneously, it allows us to be able to fine-tune things a little bit better and see the changes occur at a slower ramping up or ramping down. The last type of communication, somewhat similar to direct communication and paracrine communication, is synaptic communication. This is with the neurons. So a synapse is almost always there's a neuron talking to another neuron, a neuron talking to a gland, or a neuron talking to the muscle. So we've already talked about one of these synapses with the neuromuscular junction. Like paracrine, molecules are released from the neuron. All right, so it's not released from just any cell. It has to be released from the neuron. And the molecule released is, because it's made in the neuron, it's known as a neurotransmitter. But like paracrine, the neurotransmitter is released into the extracellular environment. Now, we try to assist that it gets to the target, because the extracellular environment is usually very small, because the neuron tries to put its axon terminal site as close as possible to the target. So the neurotransmitter has a small distance that it needs to travel to get to the target cell. Now, does some of it end up kind of missing the mark and going to other cells? Yes. Does some of it kind of turn around and try to interact with the neuron again? Yes. But the majority of the molecule is hopefully interacting on the target, and the target cell, like we saw with the motor end plate, is going to try to help itself by putting a lot of the receptors for that neurotransmitter on its membrane nearest the axon terminal. All right? So this, again, is immune. This is, in many cases, muscle. This is what we're going to focus on in this chapter, and then this type of communication we've seen with somatic motor neurons talking to skeletal muscle, and we're going to talk more about it and the different neurotransmitters that come into play between neurons in our neuron section next block. Okay. Now, what are these molecules that can be paracrine factors, neurotransmitters, and hormones? All right. They're molecules, so they can be from a variety of background categories. They can be the amino acids, so there are 26 amino acids if I remember correctly, alanine, tryptophan, tyrosine, glutamate. Any one of those amino acids can be modulated and tweaked a little bit. Maybe the carboxyl group is taken off. Maybe the amine group is taken off. Maybe something is added, so it's no longer really an amino acid. And if we modify the amino acid, then we get a molecule that technically is no longer an amino acid, and it's not a protein because it's not 2 or 3 or 4 amino acids binded together in peptide bonds, so it's an amino acid derivative. And amino acid derivatives are going to be some of the hormones and neurotransmitters that are produced by the body, because they are stable molecules that can very easily be made from a source of amino acids that we have in plentiful supply typically. And they can very quickly, as a stable molecule, be released from a cell by exocytosis, travel the synapse, travel a small distance, or travel in the bloodstream from one area of the body to another and interact with cells by interacting with certain receptors and triggering events in those cells that can have very quick effects, or in some cases for hormones, more controlled, slowly acting effects. Okay? Peptide hormones are when we start, again, putting amino acids together. So they have the GLY as one of the amino acids, LEU, PRO. Those are all just the abbreviations, and if you go back to your micro and your genetics classes, for different amino acids. And through making the message RNA and then translating that message RNA, and the tRNA reads a codon with the SANTite codon and brings an amino acid, this is the amino acid chain that comes together. And so peptides are going to be, again, small little, usually it's less than 200 amino acids. So they come together, they form a protein, but it's a very small protein. And we see this, again, in some of our hypothalamus pituitary glands that are small molecules that can be quickly made, and a lot of them can be dispersed so they can travel long distances and have effects. More advanced peptides are going to form more complex protein shapes. So if it's over 200 amino acids on one chain, and then in some cases multiple chains that come together to form a quadrary, a 4-degree structure. So what it's showing you here is this potentially is, maybe this region is one protein, and then maybe over here is a second protein, and over here is a third protein. So there's actually maybe 3 chains here, and they form their alpha and beta, their secondary structures, and then they come together and form their third structure, and then they mash the 3 of them together into this quadrary structure. And so proteins, again, they're going to be huger, bigger molecules. They're still made up at a base level of all of these different amino acids in a certain combination, but they are going to be able to travel in the bloodstream, travel like a paracrine factor, a short distance, and they are going to influence cells that they come into contact with that have receptors for them. Okay. Now, proteins, peptides are made through transcription translation. So you can only make peptides and proteins if you have the gene embedded in your DNA. Amino acids, you don't make amino acids in our body. We acquire most of them from our diet. So as long as we have an enzyme that can modify an amino acid, we can then make these different amino acid derivatives. The third category of hormones derive from a fat molecule, and so collectively they're known as steroid hormones. And steroids can come from the base molecule arachidonic acid, which is a fatty acid, and it's usually got 18 carbons that are bonded to each other. Or it can come from a different fat molecule known as cholesterol. And cholesterol, I don't remember exactly if it's like 36 or 38 or 33 carbons, but it's a lot of carbons, and they do form these ring structures. And cholesterol can be kind of snipped and modulated and, trimmed up a little bit and form a variety of different steroid molecules. And some of those steroid molecules that come from cholesterol are your cortisol, hydrocortisone, cortisone. Those are the glucocorticoids that you'd get from the adrenal gland. Aldosterone comes from cortisol. All of your androgens, so testosterone, hydroxytestosterone, testosterone, and then all of your estrogens, so estradiol, estrogen, hydroxyestrogen, and then all the progesterones. So cholesterol and lipids provide us a large number of different cholesterol-derived molecules that are known as fat-based hormones that travel in the bloodstream, and because they're fat, they have to have help, so they usually have binding proteins to help them travel in the blood, in the water, because they're hydrophilic, they don't like to be in water, and they can then travel from their target, or from their home-based organ to their target organ and have their effect. Okay? All right, so the takeaway, classes of hormones. We get hormones from manipulating and snipping and making a few changes to some of our amino acids. So those are the amino acid derived ones. We make hormones from our genetic DNA being messaged and then translated into a protein. It can be a small protein, so we can just call it a small peptide, or it can be a very complex protein that has 2 or 3 or 4 amino acid chains that come together to form a very unique structure. And then the final category are our fat-derived. So it comes from either, again, a fatty acid molecule or a cholesterol molecule. And because it's a large carbon molecule, it's typically hydrophobic. So those fat-based hormones are going to need to be bound to something in the bloodstream to help them hitch a ride in the bloodstream, because fats do not dissolve in water very easily. Okay? So let's talk about the amino acid derivatives. So the main 2 amino acids that we derive some important protein or some important hormones from are tyrosine and tryptophan. All right. Now tyrosine looks like this. So this is the base molecule for tyrosine. All right. The central carbon is attached to an amine group. The central carbon is attached to a carboxyl group. This is normally the part of amino acid that's known as the R group. So when you take chemistry and they talk about amino acids, this is the part of the molecule of the amino acid that's different. And in tyrosine's case, there's this CH2, this ring, and then this OH, this hydroxy group. And it's this combination attached to this backbone carboxyl center carbon amine that makes this a complete entire molecule of tyrosine. Okay? Now, when I do something to tyrosine, so in some cases I'm going to add another OH. In some cases I'm going to manipulate the carboxyl group and take away the oxygen, right, or add the OH. I am going to get a variety of molecules, okay? And so the first thing doing, taking off this OH and adding a hydroxy group, I can end up with the molecule that derived from tyrosine known as dopamine. Then I can add back the OH on this simple carbon here, so I took out the whole carboxyl group. I can get norepinephrine. And then if on the amine group I add here a methyl group, I can get the molecule epinephrine. Okay, so collectively the molecules or the hormones, the neurotransmitters, because dopamine and norepinephrine are made in neurons, they're neurotransmitters, norepinephrine and epinephrine are made in the adrenal gland. So collectively tyrosine gives rise to a group of signaling molecules that we call catecholamines. And catecholamines in the hormone sense of the term are norepinephrine and epinephrine, and they're made in the adrenal gland. And most of what's made is epinephrine, so 80% of what is made for catecholamines from a hormone endocrine system is epinephrine. Norepinephrine, because it's the step right before you make epinephrine, is 20%. And this is our hormone catecholamines. Now technically speaking, norepinephrine and dopamine, because we know dopamine with the shaking thing, Parkinson's is a big player. These are going to be neurotransmitters as well. So more than just the adrenal glandular cells are going to make catecholamines, okay? So they're made in neurons and they're made in the adrenal medulla, the center part of the adrenal gland, right? Now, they are one type of amino acid derivative, right? And they're very important because norepinephrine and dopamine play a big role in a lot of neurotransmitting, autonomic nervous system, sympathetic stimulation. And epinephrine is the continuation of sympathetic, so when we have a fight or flight response and we need to run away, well, if we need to run away for 2, 3, 10, 15, 20 minutes, epinephrine helps us sustain that production. Now, tryptophan, we sometimes hear tryptophan with turkey. So like this is why at Thanksgiving or after a heavy meal you want to go to sleep, especially if the heavy meal had a lot of protein, a lot of meat products in it. Because meat products have a lot of proteins, and proteins contain a lot of amino acids, and tryptophan is an amino acid. So tryptophan is an amino acid that when it's clipped and snipped in a certain way, it gives rise to the hormone melatonin, right? And melatonin, if you'll recall, is what some people take to help them sleep when they do time zone changes. So melatonin is one of the key hormones for humans that gives us our circadian rhythm, so we know when to be awake and when to be asleep. In animals, it not only helps them with their day cycle, it helps them with their seasonal cycle. So as the days grow shorter, less than 12 hours of sun, they know it's getting to be winter, and they know to grow their hair a certain way, and they know to do their behavior a certain way. And then as it gets to 12 hours of light or more, they know it's more of summer and springtime, and so they know to then do their other behaviors and slough off their hair, and it influences their breeding season. So melatonin is very important in animals. In humans, we can manipulate it somewhat with light, UV light, so we're less dependent on this as a hormone. But the takeaway is amino acid derivatives, they are going to give rise to a lot of neurotransmitters, but in the hormone world, 2 amino acids give rise to 2 categories, melatonin from tryptophan, catecholamines from tyrosine. Okay? Now, our peptide hormones, again, the key is normally it's something, peptide and proteins are interchangeable words. But for the sake of the categories here, if the Hormone is a amino acid chain, and it's less than 200 amino acids, it's going to be a peptide. If it's more than 200 amino acids, it's multi-chained, multi-complex, it's going to be a protein. Okay? So it's one chain of 200 or less amino acids. Right? The amino acids come together because the message RNA is made from the DNA, and that contains a code within the message for a protein amino acid order. And then at the ribosome, tRNA looks for the start codon, and then from there starts bringing amino acids based on the reading of the 3 nucleic acid combinations. And the codons and anticodons come together, and a certain pattern and a certain order is put together for the amino acid chain. Okay? Now, we saw oxytocin. Another small peptide is antidiuretic hormone, also known as vasopressin. They're going to be somewhere of an active protein is 8 to 10 amino acids. Now, it's not very efficient to make a message and a transfer R, and then to translate a message, it's only 8 to 10 amino acids. And in many cases, the small little protein peptide we're making, we don't necessarily want it to turn on until it's released into the extracellular area and is traveling towards the cells, traveling towards the bloodstream, and traveling towards its target. So in some cases, when you make a protein through translation, you actually have a few extra amino acids to it. So typically the start codon always does the protein I forget what it's called, but it's MET. MET is the abbreviation. And so every protein that's translated starts with MET for the first amino acid, and it's called the methylene cap. Well, you can look at this protein here, and the first protein amino acid is PYR. That's not MET. So somewhere MET and maybe 1 or 2 other amino acids are cut off and removed from the active protein. And that's what they mean when they say things are synthesized as a prohormone. Typically the methylene cap and maybe 1 or 2 amino acids are going to be cut away, liberating the rest of the protein amino acid chain that's going to be the active protein. Okay? Now, some of these small proteins are going to have a carbohydrate of some sort added to it. Now, a carbohydrate, you know, we usually think of glucose, which has 6 carbons, okay? Fructose is another carbohydrate that's 6 carbons. Galactose, lactose, maltose, there's more carbons, all right? They're small molecules that usually have carbons, hydrogens, and oxygens in a certain ratio, right? And so a glycoprotein is going to be, again, some small amino acid chain, but attached to it is going to be some type of carbohydrate molecule, all right? And some examples of the proteins that fit this category that are glycoproteins are thyroid-stimulating hormone, luteinizing hormone, and follicle-stimulating hormone. And what the takeaway is here is all of these are produced in our anterior pituitary. So typically glycoproteins are made in the anterior pituitary in the brain, and then the target for thyroid-stimulating hormone is the thyroid gland. The target for the luteinizing and FSH in females is the ovary. In males, it's the testes. Okay? So influencing metabolism, so reactions in the body for building and breaking molecules. Regulating fertility, regulating the cycle, the monthly cycle of female and males. Okay, so larger, again, different peptide hormones that are going to continue to be small proteins that have, again, 9. So the prohormone might have started out with maybe 12, 15 amino acids. A few of them are cut off, liberating ADH or oxytocin. So you see in this case, the part that's cut off is a back end plus maybe earlier the methyl cap is cut off. Okay? And what we're left with is these molecules which are going to, you know, give us vasopressin. All right? Other small proteins that are, again, about 200 molecules or 200 amino acids or less are growth hormone and prolactin. And these, like TSH, LH, and FSH, are made in the anterior pituitary. Okay? So far we've seen catecholamines come from the nervous system as neurotransmitters or the adrenal medulla of the adrenal gland for maintaining sympathetic autonomic output from the sympathetic system. Small peptides typically come from the hypothalamus and the posterior pituitary. Glycoproteins come from the anterior pituitary, and and 200 or so amino acids, or close to 200 amino acids, but still classically a small peptide, is going to also come from the anterior pituitary. Okay? All right. Moving to lipid derivatives. So if I derive my hormone from a 20-carbon fatty acid, that is going to give me an echinosoid. All right? Now, a fatty acid, you can kind of see here. Normally the carbons are made, they're not rings per se. They're usually these chains of carbons that are, you draw as like a wave, and that every point, so carbon, 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 carbon. Sometimes there are double bonds between the carbons. Sometimes things come off the carbons like they show you. But for the most part, the base molecule is 20 carbons in a row. Okay? And then what we do to those 20 carbons is, one, we usually make it 18 carbons, and that gives us what's known as a base molecule that we then can add additional pieces, make additional turns and curves and changes to the original molecule, and it's going to give us rise to a variety of base molecules that derive from that 20-carbon arachidonic acid. All right? Now, most of these molecules, so prostaglandin, thromboxane, leukotriene, lipoxin, they are important signaling molecules in your cells communicating to the immune system, and the immune system communicating to cells, and the immune system communicating to other immune cells. So a lot of what the molecules of echinosoids are going to do is going to be involved in the immune system. Now, some of the molecules that are released need to travel from, if I have a cut in my finger, they need to get into my bone marrow to tell my bone marrow to make more white blood cells, because I'm losing white blood cells as I cut my finger, and they have to go take care of the mess. So technically speaking, some of these prostaglandins, leukotrienes, thromboxanes can travel from their target cell, which might be located in your skin, might be located in different areas of your body, and they have to travel to the bone marrow, which means they have to get into the blood supply. So technically speaking, they could be a hormone because they are going to travel in the blood supply from one location where they originated to a target, which is maybe the hematopoietic stem cells, to drive those hematopoietic stem cells to divide so we can increase our numbers of white blood cells. So that's why they sometimes are known as hormone-like, but most of the time when we think of echinosoids and their effects, all of these cytokines, all these different molecules that come from arachidonic acid typically play a role in inflammatory local reactions that lead to swelling and redness and heat at an injury site, and so they usually have more of a paracrine effect. Okay? The other form of fat-derived molecules that can signal cells in different locations and around the body are your steroid-derived from cholesterol. So here's cholesterol. So it usually has those 4 rings, A, B, C, D, and then it has a few additional. And I can't quite read the numbers, but it looks like there's 29 carbons maybe. I can't quite see. All right, but what happens to this backbone of cholesterol is, just like what we did with the amino acid derivative, is we can add items and take away items, and we can start to ABCD, make it a little different than what it originally was. So in estradiol's case, the A molecule starts to have, the ring starts to have some more double bonds and has an OH still on it. But some of the others, we manipulate and take off other things and add other things. And so we can derive, I mean, look, progesterone, testosterone, estradiol, cortisol, and aldosterone. Those are 5 hormones made in a variety of locations. So adrenal cortex, ovaries, testes, some testes, you know, so they can be derived in a variety of places. Okay. The key for most of these cholesterol-derived molecules is they're very rich in carbons and hydrogens and oxygens, so they are very hydrophobic. So they do not, if you tried to put them in water, they, like oil, are going to kind of make a little oil ball that stays separate somewhat from the water. So in order to get them to travel in the blood supply, which is water, they need to be bound to proteins that help them stay in a dissolved state. And so they have traveling buddies, binding proteins, and albumin made in our liver is a major binding protein. I don't know why I'm going so fast. All right. So let me make sure I didn't miss something. All right. So secretion and distribution of hormones. So hormones like the small peptides, the small amino acids, the glycoproteins, the big peptides, the big proteins that are not bound to any kind of binding protein are known as free hormones. So insulin, glucagon, growth hormone, LH, FSH, if you look, they are free hormones. They don't have to have any help being dissolved in water. So when they are released, like over here showing, they're released from their target cells, they can travel in the bloodstream very easily because they can be dissolved in water, and then they can get to their target cell. And most of the time, these hormones that are free in the bloodstream will have effects that can last anywhere from an hour or less to maybe 2 or 3 hours. So if we think about insulin, usually when you eat a meal and then you check your blood sugars to see if insulin is working or not, and if insulin is driving glucose into the cells, you check an hour to 2 hours after your meal. All right, so what does that tell us about insulin? If insulin is going to drive glucose into the cells, it's going to do so within an hour of eating or at most 2 hours from eating. All right, now the hormone that doesn't get to the target cell, some of it, again, because it's not 100% every time it goes through the heart, going to, you know, end up in the right fork of the carotids or the right area coming off the aorta to be able to make it to their target. So some of the molecules that end up in the liver and the kidneys are destroyed, and that's one of the reasons why the endocrine cell doesn't just secrete 1 or 2 molecules for hormone signaling, it's lots of it. Okay, and then other molecules can be broken down at the target cell when they attach to the receptor. The cell can start to, after they have their changes and effects that the receptor interacting with the hormone has upon them, they can maybe be endocytosed and then destroyed within the cell. Okay, some like acetylcholine, remember there's enzymes. So different proteins live inside the extracellular fluid near the target and break up and take apart the hormone within the extracellular environment. Okay. Now, steroids and thyroid hormones, and thyroid hormones are derived from another amino acid that that makes it 2 benzene rings kind of come together. So it's a very carbon-rich molecule. They are hydrophobic, so they don't have the ability to travel in the bloodstream without help. So very few molecules could do it, but not enough to have an important effect on cells, on the targets. So most of the hormone is actually bound to special targeting binding proteins, and the binding proteins help the molecule in their travels in the body waters and in the blood supply. Now, this molecule has to eventually separate from its binding protein, and then because it's lipid soluble, it can usually cross into the cell across the membrane because lipid membranes will let lipids in. So this molecule is going to have a longer, slower effect because, one, the hormone is not always able to separate very easily from its binding protein, and then 2, its receptor is inside the cell, so it has to cross into the cell, get to its receptor, and then go and make DNA be expressed and make messenger RNA, and the messenger RNA has to be translated and then translated to a protein, and the protein then has to be, you know, ready to have an effect of changing what's going on in the cell. So these effects are slower acting, and again, the hormone is somewhat always in some level of circulation. So if I ever have or need more of it to be available to the cells, I somewhat have a storage supply, a reserve that's in the bloodstream all the time available to me. Okay. All right, so let's talk more about hormone receptors. So for those proteins, amino-derived molecules, most of them being water-soluble are not going to be able to cross the plasma membrane of the cell. So in order for them to have an effect inside a target cell, they need to interact on the membrane with a receptor. Receptors are typically a protein of some sort that can embed into the plasma membrane area, and they can be connected to other intracellular proteins, or they can have other components and subunits that are attached to the intracellular part that might have effects on the intracellular activity of the cell. All right, so if you look at it, some of our proteins might just be a little peptide that just singly transforms across the membrane. Some might be 2 peptides come together that have parts that are hydrophilic that can flow in between the fat-rich part of the membrane, but then has components inside and outside to bind the receptor and then have effects inside. Others might have multiple parts that cross into and through the membrane, and the outside of the cell part creates an area for binding its molecule, and the inside part has some ability to interact with other proteins and cause changes in the cell. Okay? Now, if you have a protein receptor, it's because you have the gene for that protein receptor and you're expressing that protein receptor. So my target cells for certain proteins are only going to be in a certain region because that's the only place that the DNA for that receptor is expressed. So let's look at like prolactin. Prolactin doesn't make milk in every cell of our body. It only makes milk in the mammillary glands of typically a female, because the mammillary glands with high levels of progesterone grow. And then they start to express prolactin receptors. And under the effect of prolactin interacting with those receptors, those glands then start to make milk. So in males, could prolactin be released? Yes. But if it's released and the male doesn't have any receptors for prolactin somewhere in their body, will prolactin have any type of effect? No, in theory. Okay? So cells that are going to have an effect for insulin have to express the insulin receptor. Cells that are going to have an effect to norepinephrine or epinephrine, because they have beta or alpha or adrenergic receptors, are going to have to express that protein receptor on its membrane. Okay? Now, the lipid membranes, remember the lipids, they can cross the membrane because they are hydrophobic. So if they can separate from their binding protein, they can go inside the cell. So their hormones that are lipid are going to have their receptors not on the membrane of the cell, but actually maybe inside the membrane of the cell. And typically that protein that's inside the cell, that's receptor, when it binds to the echinosoid or when it binds to the steroid hormone, that now new molecule of fat and protein together can go into the DNA of the nucleus and trigger gene expression. So that's part of the reason why it's sometimes called a nuclear receptor, because eventually it's going to go into the nucleus when it's bound to its hormone. Okay? Now, let's go back to these hormone receptors on the membrane for proteins, for amino acids, for things that are going to use a membrane-bound receptor. So the hormone itself never actually gets into the cell. It binds to its receptor. And upon binding to its receptor, it has to trigger events inside the cell to then have an effect in the cell. Now, the receptor is going to, in many cases, be the first part of the cascade of events that are going to take place leading to the cell changing its phenotype, changing its genetic output of proteins and molecules and activity and making or breaking molecules within. And the receptor, though, might need to take the hormone that's being attached to it and not just do one thing to the cell, but maybe it needs the cell to make more proteins from DNA. Maybe it needs to put more things on its membrane to make the cell more responsive to other chemicals. And maybe it needs to alter fuel utilization within the cell for making ATP. So to do that, the hormone is one molecule. The receptor is one molecule, but it needs to get 3 different pathways or cascade of events to start up. So the hormone receptor interaction is going to try to trigger what's known as little worker bees, little ants that are going to be able to go and initiate 3 separate cascade of events. And these little worker bees or ants are known as second messengers. So they are inside your target cell. They are, in some sense, activated by the hormone and the receptor interacting, and they are going to be amplifying. So they are going to be, if there's one hormone and one receptor interacting, you might get 20 or 30 second messengers activated. So you can have 10 of them go turn on gene expression, 10 of them alter how fuel is being utilized, so whether I use glucose or fat to make energy primarily right now in the cell, and then another 10 or 5 might go and manipulate something on receptors in vesicles being put on the membrane so the cell is more receptive to bringing in more fat or bringing in more glucose. Okay? And so different receptors for hormones use different second messengers. And the 3 big, well-categorized second messengers commonly used by many of the hormone receptor interactions in the classical endocrine system are second messengers of cyclic AMP, cyclic GMP, and calcium ions. Okay? Now, I don't expect you to be an expert in how a ADH or how a parathyroid hormone leads to cyclic AMP activation. Right? I don't need you to know every single specific terminology part, but just to give you a gist of it is when calcitonin, parathyroid hormone, FSH, LH, TSH, glucagon, epinephrine and norepinephrine interacting with beta receptors, interact with those receptors. They typically are going to lead to another protein that likes to dock inside the cell by the receptor. They are going to turn that protein on. And when they turn this protein on, and it's known as a G protein because it's made of 3 globular proteins. So it's 3 little proteins. And what happens when they're turned on is the alpha subunit is going to go turn on an enzyme that makes cyclic AMP. The beta and the gamma subunit are going to go and do something else. And you don't need to know what that is. But the alpha subunit turns on this enzyme known as adenylate cyclase. And what adenylate cyclase does is it takes adenosine triphosphate and it takes away 2 phosphates and it cycles how the 3rd phosphate is attached to it. Again, it's chemistry, don't worry about it. But it makes this molecule with 1 phosphate cyclic AMP. And cyclic AMP, it doesn't do it once, it'll do it 20, 30, 40 times. And so all of a sudden inside the cell, lots of cyclic AMP is available, and cyclic AMP is going to turn on a lot of different molecules, enzymes, and different cascades that'll let proteins be put on the membrane so ions can come in and out more freely or in a higher load or a higher number. And all of those changes are going to lead the cell to being a different responsivity, different phenotype, different metabolism going on. Okay? Now, some interactions with receptors lead to increases in cyclic AMP or cyclic GMP or calcium ions, but other interactions are going to lead to the decrease of cyclic AMP, the decrease of cyclic GMP, or the decrease in calcium ions. So some receptor hormone interactions are all about increasing second messengers, and then there has to be the opposite if you want the opposite effect, that some of the hormones interacting the receptors will decrease these second messengers and therefore take away that response that is being told originally by these other hormones. All right? So in a sense, the example is epinephrine and norepinephrine, when they use beta receptors, increase cyclic AMP, but sometimes if they interact with alpha receptors, they decrease cyclic AMP. All right, so this is why norepinephrine and epinephrine in the blood supply constricts vessels. So it constricts, it makes smooth muscle constrict, but in the airways, it causes the smooth muscle around the bronchioles to dilate. So smooth muscle is the location in the blood and in the airways, but because one is going to have an increase in cyclic AMP and one has a decrease in cyclic AMP, the effect is one time smooth muscle constricts, the other time smooth muscle dilates. Okay? Calcium, again, is another potent second messenger. Again, it's a different hormone, a different G protein, a different kind of effect that leads to calcium coming in high numbers. Calcium finds this protein known as calmodulin, and that has an effect on changing the dynamics of what the cell does. Okay? Now, the takeaway for you is that, one, hormones that can't, are hydrophobic, so they're not fat soluble, they can't go inside the cell. So in order to have an effect in the cell, they bind to a receptor membrane that is going to trigger not just one molecule inside the cell to have an effect or a change, but a variety of second messengers are going to come about and get activated, and they are then going to be able to do a variety of changes in the cell that lead to the whole cell having a change in phenotype. And that change in phenotype might be that that cell goes through mitosis and starts progressing to become an egg cell in a female. Or it might be that those cells go through mitosis and get ready to grow and make the body and that area of the system bigger, so you are growing in response to growth hormone. Okay? Now, again, steroid hormones, they are hydrophilic, so they can cross the plasma membrane, so their protein receptor, glycoprotein receptor, their molecule that binds them is going to be somewhere in the cytoplasm. And they're called nuclear receptors because typically the mojo, the cascade of events, is once the steroid and the receptor bind, they now can go into the DNA nucleus-rich area and turn the DNA on and get a message RNA made that codes for maybe one or two proteins, and that then goes to the ribosome and gets translated, and then that ends up potentially being another protein inside the cell to make the cell more able to do something because it has more enzymes, or it can be a protein that needs to leave and go have another effect somewhere else. Okay? You'll see that there's no second messengers here. So another reason why the effects with steroids is a slower effect is because the effect of steroid hormone and receptor isn't generating 2 or 3 or 4 different cascade of events. It's only generating one event. And that might be one gene being turned on, making a few maybe proteins, not a lot, and so the effects are smaller scale, smaller changing. Okay? So at this point in the chapter, we are basically just trying to get our head around what are hormones versus neurotransmitters versus paracrine factors. What hormones in the classical endocrine system, what type of molecules do they come from? Are they all proteins? Are they all derivatives of amino acids? Are they all fats? Or are they a little bit, depending on, the molecule, they can be a variety of different molecules. Okay? And then because they come from amino acids or hormones or peptides, proteins, or lipid backgrounds, what does that mean for their actions? Do they have to be bound to travel in the blood supply, or can they be free? And if they're bound, how do they have an effect, and is the effect as fast as free molecules or slower? And then when they have their effects on their target, because again, it comes back to the core molecule, is it a fat-friendly or a fat-hating molecule? Does the receptor have to be on the membrane, or does the receptor have to be inside the cell? And then when the protein and the amino acid in many cases can't get into the cell, its receptor is on the membrane, how does it then trigger not just maybe one change to the cell, but 2 or 3 or 4 things to happen to the cell when it never physically goes inside? And that's the use of second messengers. Okay? So that's basically the summary, the start of this. From this point on, we're going to get into now the specific glands, the specific hormone